Um, Tonight I'm going to talk about how genes can influence or shape behavior. And a lot, sort of as a um, background, and then to bounce off, we're then going to talk about, in general, how environments can affect behavior, and then work up to sort of cultural evolution. This is as much a history lecture as a, new stuff lecture, because this is, anyway, because, well, people argue a lot about nature and nurture and how much behaviors are determined by genes. So I thought I'd do a lecture on how genes can influence behavior, although this is going to be mostly in animals, partly because that's where you can actually do science. So I'm going to show a bunch of things, how comparative methods and phylogenies show that behaviors evolve. In other words, many examples of more taxa that share a common ancestor more recently, also sharing more behaviors. So if behaviors are often conserved phylogenetically, um, that means that they are affected by genes, just like morphological, shape traits are often shared phylogenetically. Although those phylogenetic analyses can then also show how behaviors are evolutionarily flexible. In other words, the whole point of comparative methods is to find out where similar traits evolved due to a selection pressure that we understand, not just because they descend from a common ancestor. Okay, and then I'm going to sort of go through a lot of the examples that were found in the Jacob and Goodenough books chapter, although sometimes some different ones, to look at the different methods for studying the effects of genes on behavior, inbreeding and inbred lines, selection, induction of mutation. That's something that you see in Drosophila especially. And then some more modern approaches like quantitative trait loci, knockouts, and microarrays that have been used a lot to study, especially behavior in honeybees, in everything, but in honeybees. So there are different ways that genes can affect traits. And as we know, a lot of the variation in all traits we're understanding often isn't because of differences in proteins, but is caused by differences in gene expression. So that's being studied with behavior as well as it's being studied in everything else. So that's going to be sort of the whole lecture. So we'll start with the comparative stuff. And so a classic example are the studies of courtship in Anseriformes, and the Anseriformes are, as all the bird watchers know, ducks and geese. And it's not going to be live here because this is a PDF, but those links in the PowerPoint are links to videos of different courtship behaviors. But I also have a nice one here, so I have it all queued up and everything. So here we go. Now that I've learned how to share things with sound, everything is, the world is my oyster. Okay, but this is a window that I'm going to have to expand once it starts. It's the Lab of Ornithology. So I'll copy this link into the chat. Play. Head pumping. Head up, tail up. Head up, tail up.
Sir, we can't see what you're showing us.
You can't.
No, we only heard what you were describing.
Oh, it's a scene of indescribable beauty. Those were king eiders. So can you see it now? Okay,
No,
let's see. Mother Hucker. I'm going to try sharing it again. I did not say an obscenity. I shared the name of my intramural frisbee team, the Mother Huckers, because huck is a long distance throw in frisbee. Um, okay, I'm going to try one more time. Okay, can you see it now?
we see the window. Yeah,
See the window. Okay, I'm going to start it up again. So is the problem when I went full screen?
I think so.
Okay, so
Try
this is
again.
well, maybe I shouldn't do full screen.
Yeah, it's the full screen, I think. It went away.
Full screen kills it. Okay, so
And
I
no
guess just
sound
keep it,
either.
maybe
you
just
know,
keep it in the small
I'm going to
screen.
just keep it in the small screen because
Yeah.
watch on YouTube. So, it's not giving me the choice to watch on YouTube. That's the frustrating thing. So I'm going to first, I don't know. I'm
You
going to
can
do,
you can copy the link and put it on
I,
YouTube.
oh, here we go. Now we're talking. Wait a second. It turns out I am paying too much for my auto insurance. Wait a second. I'm going to try one more time. Um, share application screen. And we're going to try the courtship behavior, which is now straight old YouTube.
Okay, this is good.
And I bet if I do this, it'll be better. Okay.
Okay. Just no sound.
No sound, and that's okay. Um, because it's saying head up, tail up. It's saying the behaviors. And even though some of them are like grunt whistle is a sound, a lot of these are not particularly associated with sounds. So pushing, reaching. And the thing is that each one of these sounds, each one of these behaviors are actually ones that were described by Lorenz. He did painstaking recordings, descriptions called ethograms, where you describe basically every behavior that a given species can produce. And then we'll talk about it. Is this bridling? I think that's bridling. I love that.
Is this a behavior for a particular reason or they're just
These are different behaviors used in courtship, so mostly by males to either attract females or fend off other males, but they're displays. And that's the interesting thing. So you can see the males will be doing these behaviors around females. In these mergansers, the males have the white wings and the green heads, and the females have sort of brown heads. So there's a female on the left, and then there's a pair. So the female's doing the behavior, then the male's doing. Anyway, so these are courtship behaviors, courtship rituals. So what? Uh-oh. And that's why I don't like doing it straight on YouTube. There you go. Okay. Right. No one can see because I'm trying to return to the slides. Okay. Okay. So what Lawrence would do is he would carefully observe, codify, name all the courtship behaviors he saw in 40 different species of geese and ducks, and then he'd compare the lists of behaviors among different species And then what he did was he assembled a phylogenetic tree. It's not going to look like the ones you see in the textbook. So in this case, mallards show behaviors including grunt whistle and bridling. We saw both of those behaviors before. And here's a gadwall, also showing the grunt whistle behavior. These illustrations are from a book and a Scientific American article that he published in like the 50s. This is green-winged teal, also showing grunt whistle. So grunt whistle is pretty widely shared. And then what he did was he built a tree based on the numbers of shared behaviors. And so if a behavior was shared by a large number of birds, it was expected to be somewhat ancestral. And birds that shared behaviors that like no one else did were deduced to be more closely related. So you can see like here's bridling, right? And bridling is a behavior shared. Oh, I've got to remember don't ever do that with your finger. Bridling is a behavior that's shared by both teals and mallards, but not gadwalls, right? So teals and mallards in this tree are grouped and deduced to be more closely related to each other than either are to gadwalls. And he built this entire tree, published in 1941, based only on the mating behaviors. So, like head up, tail up is another one that we saw. And what was really cool was that decades later, when other traits actually, even in the 40s, there was a good correspondence between the phylogenetic tree based on the behaviors and ones that were based on sort of more subtle skeleton and bone features. But then later on, when you could do either egg white protein hybridization and then ultimately DNA sequencing, there's a really good correspondence between the phylogeny that Lorenz built based on the behaviors and an actual genetic phylogeny. So that was the take home of Lorenz's, duck phylogeny is behaviors are conserved phylogenetically, just like morphological traits. So they must have a genetic basis, just like morphology. So that's the take home from Lorenz. But then the interesting thing is we also now that we can see now that we can build phylogenies that are pretty robust based on large numbers of genetic traits, we can then use the phylogenies to test hypotheses about the evolution of traits, especially are traits easy to evolve or hard to evolve? And then the other sort of behavior evolution question is, have traits evolved multiple times in response to a single like hypothesized ecological constraint? So I'm not actually going to do an example of that. I'm going to do the how constrained are traits behaviorally example. And this was looked at in shorebirds, which are a great group to study because there's a lot of variation in mating systems in shorebirds. And when we say mating systems, mating system is basically how how pair bonding takes place. And so pair bond, mating systems can be monogamous, single male and female, paired somewhat exclusively, working together to rear offspring. You can have polygyny, where one male will mate with multiple females and some won't mate at all. You can have polyandry, and there are examples of polyandry in shorebirds on the next slide, where a single female will mate with more than one male. And in fact, we'll see it in Hasana's or a couple other birds, the female will lay eggs in multiple males' nests. The males make nests. More than one male will make a nest on a female's territory. Females will lay eggs in all of those nests, and then those individual males will rear the young. You also have lek polygyny, which is where males establish territories solely for the purpose of attracting mates. So small display territories. That females will visit, and they're not going to rear their young on those territories. They're not gaining anything like nutrients or parental care or paternal care. They're just getting a chance to observe a bunch of males, and then most of them all mate with one. That's the lek polygynous system. You see that in flies. That's whenever you're in the field and you see like a cloud of like a whole bunch of flies just sort of flying at each other, it's probably a lekking display. Okay, so I mentioned before Hasana's, but spotted sandpipers also show polyandry. Females will establish territories. One, two, or three males will build nests on a single female territory, mate with that female. She will lay eggs in each of those males' nests, and they will rear them. Interestingly, they have no way of knowing whether those the chicks that they're rearing are their own. Red-necked phalarope is another species with polyandry. So then this question is, how did polyandry evolve? And you could think of it in a couple different ways. Maybe it evolved once, and that would be supported if you had a phylogeny of all the shorebirds, and all the ones that showed polyandry, sometimes called, reverse role, but that's actually sort of a biased term, um, were each other's closest relatives. That would support the hypothesis that polyandry is a fairly fixed trait, or mating systems are hard to evolve. By contrast, if you have distant relatives sharing a behavior and each of them sharing polyandry with each of them sharing an ancestor with a non-polyandrous species more recently than they do with each other, then that would suggest, actually I drew the wrong line, this one should be here. Then that would suggest that it's a flexible trait. Either it can evolve multiple times, or it can evolve and be lost, right? But it's not a hard to evolve trait. And so when this was, I think this is Lars Gustafsson, when this hypothesis was tested by mapping the behaviors onto the actual phylogeny of the shorebirds, what they found was, within this clade, the phalaropes, it looks like it evolved once. Whoops, need a pen. But then it either evolved independently two more times, or else it evolved once and then was lost, right? So it appears to be evolutionarily flexible, right? Because no matter what pattern, this pattern is not consistent with reverse sex roles being super hard to be gained or lost. Because no matter what happens, it's either been gained a few times or it's been gained a couple times and lost once. When comparative biologists are, challenged with a phylogeny and the behaviors mapped, they will use the idea of parsimony often and say that the pattern of gain and loss of a trait that involves the fewest changes is the one that's most likely. But this is one of those, which frequently is the case, a trait that no matter what path is most parsimonious, it still involves multiple gains and losses. Okay, so the sort of central dogma of biology or genetics is the idea of the sort of one gene, one protein, that genes are transcribed to messenger RNA and then translated into proteins. And what we've actually learned it a long time before, but we've learned now the mechanisms. There are lots of ways in which sort of the environment will affect how often genes are transcribed to messenger RNA. Primarily, it's how often genes are expressed. So what it turns out that because there are many steps that can be affected by environment. Transcription can be affected by tags, markers inserted on the genes, methyl groups, and others. And histones can also be modified in ways that make genes easier or harder to be transcribed. There are lots of ways in which environment can affect behavior. There are lots of ways in which actually environmentally induced effects might be heritable in a way that affects gene transcription, not changes in coding sequence. Okay, so we've actually gone over this many, many times. One of the things that we've learned since Mendel, or actually since the early 20th century, really since starting in the 1960s, is that there's a lot more to DNA than the coding regions. There are there's there are cis and trans regulatory elements, either near or far from the gene being read. There are large areas of unexpressed genes, introns. There are copies of genes that are not actually being expressed, called pseudogenes. And then we know some of the mechanisms for replications and repeated insertions of groups. A lot of our genome seems to be the sort of deactivated viral or parasitic elements that inserted themselves into our genome and are still being passed on, even if they're not expanding all the time. And then telomeres, which will regulate the longevity of a cell. So we've learned a lot in the last few decades about the functions of this non-coding region, so we don't usually call them DNA anymore. Some might be protection, so if there's a frameshift mutation and all of your chromosome was coding, then every gene's expression would be knocked out. But if there's sort of a buffer between genes, then a frameshift may not lead to lack of function in downstream DNA. A lot of different forms of regulation of expression. Okay, you know what? I think I'm less excited tonight by um, how we've discovered epigenetic effects, because most of this lecture is actually going to be on coding gene differences. I'm going to say let's take a break and let's start up in about 5 minutes at about 8:06.